Okay guys, now we are entering into a new topic of solid state fermentation. So basically there are two types of fermentation. The first one is the submerged involving liquid. The second one is solid state fermentation, means that the substrate is solid instead of liquid. As you can see, the definition is SSF, or solid state fermentation, is a system where microorganisms are cultured on the surface of concentrated water insoluble substrate with a low level of free water. They still need the water, but usually they are being put in the solid substrate and they are very limited. Alright, so this is the introduction. So it is the aerobic growth, means that we must circulate the air inside the fermenter of microorganism on solid substrates under limited water condition. The solid matrix may consist of organic based material substrate, okay, for example food waste and so on, which is solid, or inert material impregnated with liquid nutrient broth. So if we use inert material that don't have any nutrients inside, we must put together with a liquid broth for the substrate for the microorganism. Much of the growth is on the surface of the solid substrate. Large surface area per unit volume, okay, which means that usually the substrate must be grinded into a small size so that we can get large surface area. The matrix should absorb water amounting one or several times its dry weight. Okay? Because sometimes the water contain the nutrients, so we need to impregnate the substrate with water. Mixture of oxygen with other gases and aerosols should flow under relatively low pressure and mix the fermenting mash. Okay? Because this is aerobic fermentation. So this is the example of the solid state fermenter. Sometimes you can see this example during the tempeh production. Okay, they grow the fungus on top of soybean and then they will put it in the fermenter. As you can see, there will be gas outflow. You will also have air inlet. Okay, so allow the air inside so they can circulate. Inoculation inlet, sometimes you can put in the microorganism through this port. Substrate, which is a solid, not liquid. You must have cooling plate as well to control the temperature. When you buy the tempeh, usually when you touch the tempeh, you will feel the heat because the fermentation itself will produce heat because it is a metabolic reaction. So we must have cooling plate. Okay. So this is basically the design of the solid state fermenter. Okay. Traditional solid state fermentation, for example Japanese koji, we can grow this koji is a fungus. We can grow them in the solid substrate such as steamed rice or on the soybean for the production of soy sauce. Indonesian tempeh or Indian ragi, okay, for the production of tempeh. We use starter culture of Rhizopus oligosporus and Rhizopus oryzae. French blue cheese with used perforated fresh cheese as substrate and selected molds. Okay, another fungi. Composting of lignocellulosic fibers, naturally contaminated by a large variety of organisms. Okay, so this is probably a mixed mode fermentation, means that not only fungus will also grow bacteria as well. Okay, so that this mixture of microorganism can compose this fiber and can be used for example the feed of animals. Okay, koji molds produce enzyme, for example this kind of enzyme, and hydrolyze starches, protein and fat into different product. Okay, so for example if you use koji on the soybean, it will start break down certain carbohydrate and protein and produce a newer one which is probably better in terms of nutritional content. Okay, after you mix the starter culture with the substrate, you will put it this way to increase the surface area. Okay, this is the example of production of tempeh from traditional. Okay, here traditional. to a better modern method, okay, in a specialized room. Okay, usually when you do it traditionally, you just grow them in the natural environment. Okay, industrial solid state fermentation means you use large scale industrial process to produce in bulk. For example, koji production has been changed to a more complex processes, creates bedroom fermentation, rotating drum, automated stainless steel chambers or tray, electronic sensors, mechanical stirring, loading and discharging. Traditional sake or miso factories is around 1 or 2 metric tons per batch. Now it can make up to 20 tons. Okay. Another application is production of pectinase from the sugar beet pulp. Okay. This is the example of rotating drum fermenter. As you can see here, this is the exact picture of the cylindrical drum. Okay, this is the representation. Rotating drum means that they have this drum structure, and then you rotate this drum. This one is the substrate, okay, usually the solid substrate, and this one is the baffles to assist in the mixing. Okay, usually you will only have around 30% filled with bag. Okay, if you use more, the mixture won't be as efficient, and the air is blown through the headspace. Okay, because as I said earlier, the air is required for aerobic fermentation. Okay? This is the tray fermenter used extensively for the production of fermented oriental foods such as tempeh and enzymes. Their substrate are spread onto each tray to a depth of only a few centimeters. This is an important point. Okay? Then stacked in a chamber through which humidified air is circulated. So we put air in and air out to allow for the circulation. The system requires numerous trays and large volume incubator chambers up to 150 meter capacity. Okay, this is bag system. The bag system is actually very similar to tray system, but it is usually used commercially in, for example, koji production, consisting of bag substrate up to 1 meter deep, which is the main differences. The main difference with tray fermenter, through which humidified air is continuously forced from below. Okay, so we force the air from below to allow the air circulation. Okay, and we have a thick layer of solid substrate bag. Okay, this is the factors that are affecting the SSF culture. We will take a look at each one. The first one is moisture, carried out with little or no free water. Instead, the water is usually in the form of the air, and the water is usually in the solid substrate, which is very low. The moisture content is 30% to 80%. Fungi just require 40 to 80% water, while and bacteria, the water activity should be no less than 0.9. That's why this type of fermentation is usually done for yeast and fungi, because they can survive in a very low water activity, which is 0.7 and above. The water activity affects growth and product formation. So we can play around with this factor to allow for different product formation from similar microorganisms by changing the water activity amount. Optimal water activity depends on agitation rate and cultivation temperature. So if we agitate more, we will get probably higher water activity, and if we play around with the temperature, it can also affect the water activity. Water activity is controlled by aeration, humidified air, and water spray. Okay, we must also control the temperature inside the fermentation tank. The cellular respiration is highly exothermic, means that they produce heat because of the cellular respiration itself will produce heat, because cellular respiration is catabolic process, and one of the byproduct is heat. And the second thing is because there is limited heat transfer capacity of solid substrate. Compared to the liquid substrate, liquid can allow higher heat transfer, but if you use solid substrate, it will allow less heat transfer. In aerobic processes, heat generation may be measured from the rate of CO2 evolution or O2 consumption. As you can see here, they calculate that 1 mole of CO2 is equivalent to 670 kilocalories, and therefore it will produce certain heat, comes together with the energy that is being produced. Removal of metabolic heat is a limitation because of the poor thermal conductivity of the solid substrate, as I mentioned earlier. Temperature control during fermentation is obtained by evaporating cooling and circulating coolant. So we need to use a certain type of coolant to cool down the fermentation to allow for optimal fermentation by the microorganism. Okay, next one is pH. Metabolic activities can either reduce the pH or increase the pH depending on one, the production of the products by the microorganism, or two, by degradation of certain compound. Okay, for example, the reduction in pH is secretion of organic acid, so the solid substrate will become acidified. The second one is ammonium salt consumption, so they will reduce. Ammonium is alkali. If the microorganism consume ammonium salt as part of their nitrogen content, nitrogen source, it will reduce the pH. Increase in pH caused by absorption of organic acid by certain microorganisms. Second, urea hydrolysis will result in alkalinization. Species of microorganism, for example, these three fungi species, they can cause the pH to drop very quickly, while these three fungal species, the pH is usually stable. Okay, so sometimes we can use the substrate that can act as a buffer. For example, lignocellulosic materials. Okay, usually the solid state fermentation are practiced without the use of pH control during the substrate preparation only. So after that, after fermentation is ongoing, we can start controlling the pH. In some non-sterile processes, the initial pH may be adjusted to pH 4 or less to prevent the bacterial contamination, because usually bacteria cannot survive below pH 4. In the media that are rich in proteins and amino acids, deamination or breakdown of amino acid may contribute to the increase in pH. Some pH stability can be achieved by using combination of urea and ammonium sulfate as carbon and nitrogen sources. Okay, these two will act like some sort of buffer. So the uptake of aminium, because the uptake of aminium causes rapid acidification. Okay, for the oxygen uptake parameters, aeration fulfills four main functions in solid state fermentation, namely two. The first one is to maintain aerobic condition, like I said to you earlier. The SSF requires aerobic condition. Second, to dissolve carbon dioxide that is being produced by the microorganism. Third, to regulate the substrate temperature. Okay, because aeration can remove the heat from the solid substrate. And lastly, to regulate the moisture level by adjusting the humidity of the air that we use for aeration. For the substrates, all substrates have common features, basic molecular structure. Okay, I will continue this one in the next lecture because the 15 minutes is almost up. We will continue with the substrates. Thank you.